Dual color or fusion fish studies. This form of testing utilizes DNA probe systems that are designed to detect well-known neoplastic rearrangements affecting two specific loci, but may not detect variant, complex or typical rearrangements involving these loci. Additional secondary clonal rearrangements may not be detected in interface nuclei. Further characterization of abnormal cell populations by metaphase fish or conventional cytogenetic methods is highly recommended. Dual color or break apart fish studies. These studies utilize DNA probe systems that are designed to detect the involvement of specific loci known to participate in rearrangements involving a variety of translocation partners. These types of DNA probe systems can only identify rearrangements involving a single locus and cannot identify other loci that may be involved in the rearrangement by interface analysis. Further characterization of abnormal cell populations by metaphase fish or conventional cytogenetic methods is recommended. Flow sorting and micro-dissection. The origin of marker chromosomes that were unidentifiable by standard banding techniques could be verified by reverse chromosome painting. This technique includes micro-dissection and flow sorting. Chromosome-specific sequences are highly desired in studies focusing on comparative genomics and genomic organization. Specific chromosomes can be isolated from the rest of the genomes by flow sorting or fluorescence activated cell sorting (FAC) and chromosome micro-dissection. In flow sorting, chromosomes are sorted using a laser system that distinguishes chromosomes depending on their size and fluorochrome affinity, that means the AT, GC base content, while in chromosome micro-dissection, chromosomes or chromosomal segments are literally scrapped and collected. In contrast to flow sorting, micro-dissection allows the small chromosome fragments. It is generally assumed that flow sorting generates panes of greater complexity and coverage because this method allows the collection of around 300 to 500 chromosomes. Chromosome micro-dissection was originally developed in studies involving Drosophila polytene chromosomes as a way to obtain DNA markers from specific chromosomal regions and was quickly applied to mammalian genomes during the 80s. Only minute quantities of chromosomal DNA are available after micro-dissection. The quantity of this DNA depends on chromosome fixation and staining. The subsequent development of in situ hybridization procedures and establishment of degenerate oligonucleotide primed polymerase chain reaction or DOP-PCR allowed the application of chromosomal probes obtained by micro-dissection in fish. It is very rapid, is less difficult to handle and involves direct PCR amplification of DNA contained in micro-dissected fragments with a universal primer composed of a mixture of oligonucleotide sequences that lacks complementarity to the target template sequences. DNA isolated by flow sorting or micro-dissection can be used for genomic research, including the generation of chromosome-specific libraries or markers, physical mapping, and cloning of chromosome-specific expressed sequences or genes.